Welcome everyone to our next module based on food safety, where we are detecting different kinds of spoilage causing organisms in food sample. Today, we are going to demonstrate how to determine and confirm the presence of aciduric, flat, sour, spore formers, specifically Bacillus coagulans, in food sample. So let's get started. Aciduric thermophiles that cause flat, sour spoilage can be identified by their ability to survive in acidic environments and produce a sour taste without causing the food container to swell. This spoilage is common in products like tomato juice, where a fruity or medicinal flavor develops and the pH of the product drops by zero point 0.3 to zero point 0.5 units. For this procedure, you will require or need the following materials: food sample that is to be analyzed, sterile test tubes, sterile bromocresol purple agar buds, sterile Petri plates, sterile pipettes, heating apparatus like a water bath, and an incubator set at 55 degrees Celsius. Let's start now. First, take weighed samples or prepare dilutions of the food samples in sterile test tubes. Now it's time to give the samples a heat shock. For this, we will place the test tubes in a water bath set to at 88 degrees Celsius for 5 minutes. This heat shock is crucial to activate the spores. After 5 minutes, immediately cool the samples. Once cooled, add 1 ml of each sample to sterile bromocresol purple agar buds. Ensure you use sterile pipettes to avoid contamination. Next, incubate the inoculated agar buds at 55 degree for 48 hours. The high temperature, that is 55 degrees Celsius, favors the growth of aciduric thermophiles. After 48 hours, observe the plates. On dextrose tryptone agar with bromocresol purple, surface colonies of Bacillus coagulans appear slightly moist, convex, and pale yellow in color. Subsurface colonies are compact with fluffy edges. The yellow zones surrounding the colonies indicate acid production. For calculation, count the number of colonies formed on each plate. Calculate the average plate count from multiple plates if necessary. Multiply the average plate count by dilution factor to determine the concentration of spores. Express the final count as X per gram, where X is the calculated spore count per gram of food sample used. Record the number of suspected colonies per gram of sample. These results confirm the presence of aciduric, flat, sour spore formers in your food sample. And that's how you can determine and confirm the presence of aciduric, flat, sour spore formers in food samples. Thank you for joining us in this lab guide. Remember, maintaining food safety safety is crucial to ensure the health and well being of the consumers. Thank you so much. This is the link of the reference.